coming to the topic of cell cycle and cell division. So usually we draw a circle like this. So we take only 95% of interphase, which is, if you take the human cell, it would be around 23 hours. And the major part will be G1 because that is a growth mainly, and this is M. So this entire thing, this is gap 1, this is S, this is gap 2. So this entire thing you can call it as interphase from here to here. It's the interphase. And this one is M phase, which is 5%, and this is like 95%. And G1 is the major, and we call prophase, metaphase, anaphase, telophase, and we forgot one thing, sorry. We have the cytokinesis. So this all together, so M phase consists of karyokinesis and cytokinesis. And coming to prophase, metaphase, anaphase, telophase, this is the karyokinesis. So if you take into partition, so cell cycle is divided into two major phases. One is interphase, other one is M phase. Okay, so coming to the interphase, we have the categories. G1, S, G2. And M phase, it is again divided into two categories: karyokinesis and cytokinesis. So coming to the karyokinesis, we again have prophase, metaphase, anaphase, and telophase. Okay, cytokinesis we have a different for plant cell and different for animal cell. Okay, so when a eukaryotic cell was being created from pre-existing cells, so omnis cellula e cellula. So the cell later divides into two cells. Of each daughter cell will repeat again and again. So one cell will form into two cells, and two cells will form into four cells. Means, so by dividing the cells, it is actually multiplying. So this is the only place where you can see division and multiplication is same. This cycle of growth and division is known as cell cycle. So it takes place in a controlled, a coordinated way, controlled by genes and factors as usual. These are actually a sequential event. If you see, it is a sequential event from the time of formation up to its own division into two daughter cells. The time required to complete one cell cycle, that is beginning of one cell division to the next, is the generation of time. So it is 90 minutes for yeast. For yeast it is 90 minutes. And 8 to 10 hours for mucosa of intestine. And human cell in culture, actually in culture, it will take 24 hours. Human cell in culture, it will take 24 hours. Though the details of cell cycle vary from organism to organism, because certain events are universal, the time will vary. So coming to the interphase, this is the interphase. You can see it is the time period between the two successive M phases. So interphase, M phase. Between two successive M phases, you have interphase. This period is active synthesis, metabolically active, but intense growth will be there. this stage is active and components will double in size. So if you take human cell, 24 hours, 23 hours will take place for interphase only, which is 95% of the duration of cell cycle. So it is again divided into 3 phases, G1, S, G2 and G0. So we'll see what is G1, what is S and what is G2. And again we have G0 also, technically which is G1, we'll see what it is. So G1, during G1, that is metabolically active and grow continuously. So it occupies the major portion of cell cycle. So out of this 3, majority will be of G1 only. It synthesizes the cell components except DNA. That organelles like mitochondria, chloroplast, ER, Golgi bodies, lysosomes. Because vacuole cannot synthesize, it is not needed. And regarding to this tRNA, mRNA, rRNA, ribosomes, all these things, the metabolic rate of the cell is very high. During this, cell will undergo structural and functional proteins also it will form. And functional proteins. During this phase, the cell will undergo the process called differentiation. Next, we'll see the S phase. So coming to the S phase, we otherwise call it as synthetic phase. Coming to S phase. So where the DNA replication will take place. DNA, histones, centrioles. So proteins associated with the chromosomes, namely histones, are synthesized, and 2 chromosomes are attached at the centromere. So particularly at the centromere. So usually the proteins called cohesins are keep the chromatid attached at the centromere. The DNA content, for example, it will be doubled. In haploid it will be 1C to 2C. In diploid it is 2C to 4C. There is no increase in chromosomal number. No number will not increase. That is, the chromosomal number before S phase is N and after S is also N. And after M is also N. It will remain as N. If it was 2N, it will remain as 2N. Centrioles will duplicate at S phase in the cytoplasm. And next, gap 2, G2. This is characterized by the synthesis of proteins. Mainly proteins are synthesized in this phase. While the cell growth will continue. So chloroplast and mitochondria will double. So and now in this, the proteins of spindle fibers are produced. And there will be active metabolism to store energy. So energy is needed for M phase, right? That is stored. So coming to the finally G0 phase, or we otherwise call it as quiescent phase. In the quiescent phase, so in which metabolically active cell do not undergo division permanently or temporarily. So the cell exists from cell cycle. In G1. So technically G0 is none other than the G1. So is differentiate to form specific functions such as nerve cell, skeletal muscle, cardiac muscle, matured RBC, WBC. Some cells in G0, if stimulated, will return back to cell cycle, like fibroblast, parenchyma, to form secondary meristems and ligaments after ligament tear. So mitogens are the substance which stimulate the cell, like cytokinins in plant cell, epidermal growth factor in animal, platelet derived growth factor for lymphokines, lectins are the things which will initiate the cell cycle. Okay, this is the things about G1, S, G2 and G0. Thank you.